So Professor Niranjan Professor is with us today. So I request Madam Subhasri Das to read a brief biodata of sir so that the lecture will be started. Please, Madam.
Thank you, sir. Namaskar. Very good morning to all. I welcome you all to the second day of our 21 days national training course, starting with the lecture by Professor Niranjan Panda, sir. Professor Panda is the Dean of Research at Orissa University of Agriculture and Technology, Bhubaneswar. He has served as Head of Department in Animal Nutrition in the College of Veterinary Science, Animal Husbandry, OUT, Bhubaneswar. He is an outstanding researcher and an
Ma'am, please unmute yourself.
Okay. Namaskar. A very good morning to all. I welcome you all to the second day of our 21 days national training course, starting with the lecture by Professor Niranjan Panda, sir. Professor Panda is the Dean of Research at Orissa University of Agriculture and Technology, Bhubaneswar. He has served as Head of Department in Animal Nutrition in the College of Veterinary Science, Animal Husbandry in OUT, Bhubaneswar. He is an outstanding researcher and an excellent teacher with more than 30 years of experience in teaching, research, and extension. He was conferred with the Best Teacher Award by Animal Nutrition Society of India in 2022 for his remarkable achievements in the field of teaching. He has been bestowed with the Fellow of Animal Nutrition Association title as a distinguished animal nutritionist not once but twice. He has contributed in more than 100 research, extension papers, and many book chapters with his vast and in-depth knowledge in his field. So he has guided more than 20 students in their successful completion of PG, PhD research and has achieved many recognitions at the national level. So I would now request Professor Niranjan Panda sir to deliver his lecture on nano mineral supplementation in poultry ration, advancing nutritional strategies for improved farm profitability. Please, sir.
Good morning, everybody. Are you able to hear me? Or Subhasri?
Yes, sir. Yes, sir. Sorry, sir. Namaskar, sir.
Namaskar. Okay. So, good morning, everybody. Thank you, Subhasri, for your nice introduction. So, pardon for some delay in this process. Actually, in my room, there was some net failure. So, I came to our Center of Wildlife Health where Dr. Korab is sitting. So, we'll start with the sessions. This is the second day. I think we have come with the first day session of Dr. Patra. So today, though the topic was there for the nano supplementations in the poultry, so thinking the diversity of the candidates and participants, I just modify my topic slightly, the application of nanotechnology in agriculture and animal husbandry. So I make a broad-based topic so that everybody can be in a better position to deal. So if you see, what is a nano, how is the nanotechnology, how the synthesis of nano can be done, what economics of production of the nano, and how it affects the growth and immunity and other things, and how we can screen an outcome and way forward. So with the broad headings, we can go for the contents. It might take 40 to 45 minutes for my presentations. So if you see, this nano was not a very new concept. Later it has been derived from 1959. The concept of nanotechnology was first proposed by Richard Feynman at the California Institute of Technology, which is known as the father of nanotechnology. So it is the word which is derived from the Latin word of nanus, that means dwarf, which is commonly defined as the combined art of science and technology of small things. So it is a now National Nanotechnology Institute. So what it defines? It defines that the nanotechnology is the manipulation of a matter with at least a size of from 1 to 100 nanometer. So this is the probable size. Nanometer means I am telling 10 to the minus 6. That means where we can go. This is known as a nano. So variation is from 1 to 100. I mean below 100, that is a better one. So this technology has a major role in the production, processing, transportation, storage, safety, and security of the food. So we will go how the nanotechnology have been applied in the field of agriculture, how we start of agriculture. So a lot of scope is there, lot of use, and you see every day there is a nanoscience application of nano is there. And you see number of journals are coming with the nano itself. More than 10 journals with the nano itself is coming. So then how much a day you are coming with the nano? So it improves crop protection, nutrient delivery, disease management, precision farming through nanosensors. So nano fertilizers, nano pesticides, and how we can easily detect the disease with the nano use, and then the food quality, safety, and finally the environmental impact. So all the things are nano we are going through the application in our agriculture is continuing. And as per the nano fertilizer pesticide is concerned, so the conventional fertilizer, if you see, we are using, we do not know how much it has been taken by the plant or how much wastage through the process of leaching or through air or water. So there is a good percentage of fertilizer, conventional fertilizer wasted. But as per the nano fertilizer concern, we are very specific, we are very using very little quantity, and it should be utilized by the crops in a very effective way. So as far as and because the delivery is slow, its availability is very good. So that's why the crop can take in the whatever fertilizer giving in a better way. As far as the nano pesticide is concerned, so nanoscale pesticide formulations and the solubility, dispersion, target specific delivery and efficiency compared to conventional. It is a very targeted one. The pesticide you are giving is a very very common one. Because nowadays when you are giving any immunotherapy, previously because the cancer patients were telling the very lot of side effects were coming with the cancer chemotherapy or radiation like this. But now we have gone a very micro level so that we can have a specific targeted way we are applying the chemo or like this so that it can effectively means it can kill the specific targeted RNA or DNA rather combining the whole body. Similarly, pesticides, whatever you are using, is a very targeted way, and this targeted specific means pathogens or parasites or nematodes or entomology. It is a very specific way it is acting. So nanocapsules, nanogels, emulsions are allowed slow and sustained release, and lower doses are more effective, and you can reduce the toxicity of the nano pesticides what are regularly using conventional. Similarly, nanosensors. We are using a very much nanosensor we are using, so we can monitor the soil quality, we can monitor the growth of the crop, we can monitor the plant pathogens, and the moisture level is a real-time basis. So means what is the real-time basis? How you can apply your irrigation, you can apply the fertilizer as a real-time basis by using the nanosensors, and also silver nanoparticles as applied in most of the coatings on the greenhouse glass, plastic films and irrigation. So it is mostly, if you see any pipe where we are coating with the nano, that means it protects from the wear and tear, and finally it can prevent the microbial buildup. Because when it is going regularly, was going through any type of substance and parting to a coverage or that is the films or a pipe. So there is a effect is there for coating is helps for prevention and wasting. So also, if you see the application of nano silver, is we in our college also we have done a lot of work on nano silver also. Department of Surgery, Department of Pharmacology, how the healing of the wound can be done not only in the humans but in the animals also. Wound healing process can be done with this. Also in water purification, there is a great role in the water purification. Then the where magnetic nanoparticles, carbon nanotubes, nanofilters enable rapid decontamination of the water from the pesticides, fertilizers, pathogens, which can safely reused for the irrigations. So rapid diagnosis of patient early disease prevention is our very similarly on the delay of ripening can be prevented. Spoilers can be done during storage process. You can do also can reduce the storage loss also. Seed germination we can do also, food packaging, then crop protection, then all the things. There is a lot of application of the nano in the agriculture and allied sector is there. So in the food processing, there is a great role of the nano is there. Encapsulation and delivery, the nano encapsulation of nutrients like vitamins, minerals, antioxidants, and flavors, in the food matrix through techniques like that is the nano emulsions, nano lipones and bilayer vesicles. So all these are encapsulation is done to prevent some of the vitamins you are giving. So vitamins might be destroyed with the evaporation, means with this process of oxidations or means when you are exposed to the air or your oxygen. So that can be done with this encapsulation process. So food safety, nano silver particles incorporated in the food containers and packaging will provide antimicrobial protection, avoid contaminations. Similarly, magnetic nanoparticles bind and detect pathogens. Most of the, if you see, 90% of the means infected pathogens is are salmonella and E. coli. So when you are we're using the nanoparticles, then we can prevent this salmonella, E. coli when it is bind with the pipes or anything we are covering. Then we can prevent this growth of the salmonella and E. coli. And nanoparticles is a feed additives. Is my role of a nutritionist. We can add the feed additives is there in animal nutrition. The nanotechnology is used in the preparation of minerals, especially trace minerals, where the bioavailability is very low. If you see the bioavailability of most of the trace minerals like selenium, zinc, copper, cobalt, all these are not that much not bioavailable in the process when we are giving to the animals or human beings. So nano minerals have a more bioavailability. As I already told, that the size is less than 100 nanometer and which can quickly pass through the stomach wall and to the body cells, reduce intestinal and mineral antagonism. Most of the time when we are giving minerals in a combination of the mineral mixture, there is a lot of antagonism takes place between the cations, between the anions. There, if any of the one of the cations are increasing, then there is requirement of other is there. So a lot of antagonism takes place inside when you are giving in inorganic form of the minerals. So that is there how you can prevent this antagonism through application of the nano and improve digestive efficiency, immunity and performance in the Ration poultry. How you can go through one of the experiment, because I was one of the PI of the project of nano selenium, and which is the innovative project from the ICR. From that we have got some funds and we have studied in 3 years we continued the project. So I will, what are the findings I can have to share with you. So food and food industry also improve functional and nutritional values, sensory properties and processes to the self-life, or just like told all these things actually. So can we synthesize? Then the question arises, can you synthesize nano in our laboratory? Yes, we can. We have the lab facility in our department also. So this can be done from chemical method. There is one green method. There is 3 methods are there. One is chemical method, biological method, green methods. So I'm describing how the nano selenium, because my concern is on nano selenium. I prepared because the selenium is such a minerals which is the limit of toxicity and its requirement is very low. So that's why if you are very low, means deficiency may occur. If you are having a low dose, if you are giving a higher dose, toxicity may occur. So, and selenium, if you know, it is one of the most important antioxidant trace minerals is concerned. It is a part of the glutathione peroxidase enzyme, which combination of the vitamin E, it protects the cell membranes from the oxidative damage. By this way, we can prevent and we can increase the immunity. That's why selenium is one of the very most important trace minerals as far as the animal and the human being is concerned. So nano selenium particles are usually synthesized from the sodium selenate. This is inorganic sodium selenate being used as a glutathione reductase and a reducing agent, and bovine serum albumin as a stabilizer. How the process is going on? This is our laboratory. We are preparing these nano selenium particles, and finally lyophilization takes place. Similarly, from the microorganisms, you can prepare the nano. That is the submerged fermentation techniques was employed were selected from the microorganisms. That is Proteus species, Bacillus, and Sporosaccina species. And this is how the process is there. We can have the inoculations, then incubation, then centrifugation, or separation. How the synthesis of is there from the microorganism. We can also synthesize the nano from this. Similarly, we can also synthesize from the green itself, in the plant itself. We can take a coriander, we can take the garlic, ginger. We can take. And one of the we found out coriander is a better substitute, better than the ginger and garlic. If you see the nano is there. So this is where extraction process is there. We are doing in our laboratory how the process of the 3 process, and out of the 3 process we have seen the characterizations we do, the chemical synthesis of nano, how we are doing, the average size we come is around 55. I've already told if it is less than 100, but the average is 55 is a better one. That in the electron microscope, we have seen what the size of the nano we have synthesized. It is around 55, average 55 nanometer. And we have also spectral analysis of the nano synthesized in vector isolates. DLS analysis we have to 10 images, all we have done how the characterizations and the nano is there as per the selenium is concerned. So this is how we have done. And if you just have a compare what is the economics of productions of the nano selenium using the three methods. So we found out that by producing the cost of production per gram of nano selenium, it is around 9,000 per gram of nano selenium is produced the chemical method by using the sodium selenate, and whereas the biological method is only 777 and green method is 690. So with this we found that green method is a better one. We are utilizing in the coriander leaves how this green method can be process is there. So just by sake we can, because here we are utilizing the reduced glutathione, it is very costly one. That's why the cost is higher is there in chemical method. So we have done several experiments with the poultry bars, how different doses of the nanoselenium starting from the 0.018 ppm to 1.2 ppm we are giving, and we are control. There's a positive control we are telling is that is without giving positive control without giving selenium, and a negative control if positive control we are giving is a inorganic selenium, that is 0.3 ppm inorganic selenium, to the standard recommended of the Bureau of Indian Standards as per the poultry is concerned, 0.3 ppm of inorganic selenium in the T2, and others we are graded level of nanoselenium from 0.18 ppm to 1.2 ppm we are giving, and the control without supplementation of any nano means selenium. So we found out that about around 0.15 ppm that the T6 found a better means a growth rate in comparison to others, and the comparison to control, and as well as the inorganic selenium. And similarly we do in the summer season, we do in the winter season, and we do in the 3 seasons we do, and almost we found that in the summer season what we have just manipulated with addition of vitamin E. We have also means we have studied in addition to the nanoselenium, that is the only 3 level we have taken, actually 2 level we have taken, 0.15 and 0.3 levels, and vitamin E 2 levels, that is 100 and 200 levels. And finally we got the combination of both the nano selenium as a 0.3 level and vitamin E as a 200 milligram per kg in feed. We found a better feed, a better growth rate in case of broiler birds. So my idea is telling, when the summer is there, when the excessive stress is there, when the birds are under stress, there is the nanoselenium and vitamin E acts synergistically as a antioxidants. So we have that is summer, okay. So we found from this actually that the graded level of nanoselenium from 0.18 ppm to 1.2 ppm were supplemented to the commercial broiler birds in 3 seasons. And it was found that 0.375, 0.150 and 0.3 ppm levels are found to be better growth FCR during winter, rainy, and September. Whereas in case of summer, we found that supplementation of nanoselenium with 0.1 ppm with vitamin E with a rate of 200 milligram per kg is a better than the other control groups. So these are the how findings were there. We have studied actually, and the project continued for 3 years. And this is the further screening was done actually, because we have done a greater level. We have later we go for only 4 doses of nanoselenium, and we found after coming up the 7 or 9 doses we are restricted to only 4 doses here, and we found that finally we found that that is in the 4th week and 5th week we are supplementing, that is a 0.15 ppm in the 4th actually or T3 we found, that is 0.15 and that is T4 and T5. We found that there is significant growth of this body weight in case of broiler birds, and the FCR also almost very good. FCR 1.5 FCR, 1.51 FCR is one of the very good FCR we have found in these doses. And the bioavailability, what is the serum selenium level, we also found it is better in the T5 and the T4 levels. T5 is better than T4, and T4 and T3 are similar, but all are very higher than the T1 and T2, that is without selenium supplementation and a low dose of selenium supplementations. So antioxidant enzyme starting from the catalase, the glutathione peroxide, we found there is a very good catalase. We do not found any difference, but as per the SOD is superside dismutase, we found a significant difference is there. As per the GPX is there, glutathione peroxide, there is also good effect we have found. So this is just a, I am giving just effect as per the immunity is concerned. We also found the HA titer value. Both these higher levels we got a better result, and the cutaneous specifically hypersensitivity and the higher doses we are getting better, and still better we are giving in the 0.3 ppm. So like this, we also whatever the E. coli, that is how much the intestinal bacterial count as per the intestine is concerned. So E. coli, if you see the increasing doses of the nanos, we found that the load of bacteria, which is the major predominant bacteria in the duodenum and jejunum, mostly in the intestine, we found that the E. coli number decreases with the increase of the selenium doses. So this is a very good positive point, that it acts on the also bacterial growth is there. Salmonella counts also the decrease is there. And reversely, if you see the positive bacterial lactobacillus, it is having a better value than the control in the smaller doses actually. So like this, due to histology, we also do the villi length and the villi width also we have studied. There is a very good dramatic increment of the villi length, and with the help of the anatomy department, pathology department, we do this actually. The genome histology we also do, how the intestinal morphology changes. That is the pathological intervention. That's why perhaps with this actually, and another study actually, is there any toxicity levels of the liver and kidney, which we see actually, because we are giving at least 0.3 level, 0.15 level, but we do not find any also abnormal hepatocytes and kidney cells in the slides. You can see there is no that much damage of the liver and kidney was there actually. So that's why. Then what you do actually, also one of the co-partner from the NDRA was there. He was also associated with the different genes and proteins associated with the stress and immunity. So gene expression we also know actually. We found out the expression of the genes and the proteomic study in the help of the NDRA. Dr. A.K. Mahanti was there, who scientist. So expression we know it's related that the two levels, 0.15 and 0.3 level supplementations have a better having that the monoclonal superacid production due to oxidative stress was there actually. It is reduced in this T7 where there 0.15 or 0.3 ppm supplementation was there. So like the gene expression study also associated with this actually, we conducted this. And finally, after the end of the we taken these three levels and we prepare the pelleted feed finally with this nanoselenium. With the last part of this is the objective was there. You prepare a feed with the pelleted feed and supplement to the birds. Similarly, we do for the with three levels we have taken, and we found that actually the three methods of nanosensory, and we found that the 0.15 ppm and 0. Vitamin E and previously slide actually sorry. So we found in the impregnated feeds, we found that there is the antioxidant status improvement, body weight gain was there, immunity, and all these things were improved with the selenium improvement feed we have given actually. So like this, finally outcome of the research was there. So out of three methods of synthesis, nanoselenium green method, the coriander we found to be economical, and supplementation nano with the 1.5 ppm and vitamin E with 100 gram in the diet, and somewhere improve the growth and immunity. And a nanoselenium concentration 0.3 ppm has significantly changed the expression of the antioxidant enzymes like SOD, CH, and glutathione peroxidase, and immunity of the birds in the reduced stress. So if you see, then I come into the very general one. Is there really we are getting benefit out of this? So there is challenges of the nanotechnology in agriculture is there. Yes, there is benefit is there, also challenge is there actually. You see toxicity concern. The impacts of the nanomaterials on soil quality, microbial activity, and human health require more evaluation through the life cycle analysis, and also trigger the production of, because most of it we have gone for a one season or one time or a one generations. We cannot continue for further actually. Further continuation should be there. Any toxic levels is there. Though you have seen effectively, further it can aggravate the to generation we are giving. Still, it is in a dark side is there. Nanomaterials reaching the land have a potential and contaminate the soil, migrate to the surface and groundwaters. So, as a natural, because it is in an iron research, there is also high cost. With this cost, if you see, if we compare the traditional or conventional minerals or conventional pesticides or conventional fertilizers, no doubt it is a very means a costly one. We have to see the cost into factor. Then the scalability, the large variety of nanomaterials are still only being produced in large quantities. So regulations, still we have not that much regulatory mechanism should be there. How to lack of standardized the safety data and non-specific regulations due to the commercializations, lack of awareness, skill shortage. So all these are there. It is really challenge for us how we can go forward. There is limited interdisciplinary expertise because nano is not a domain of only agriculture. It is in the all science in agriculture, in food technology. So in a combination way, we can see how it can be done, or how this can be applied to a human and ecology as an environmental. So all this environmental also have a great role also. So the skill shortage is there, and the application interdisciplinary approach is there. We should do when we are doing any nano experimentation. So all the things starting from the soil, starting from the plant, starting from the animal, starting from all the things in the environment should taken into account. So this is to be taken care of. And all the things also, we are also ICR also developed some nano pesticides, nano sensors. So all the things ICR also developed. IFCO also have done in the first one in nano urea and nano DAP productions. So there is a good collaboration with the different projects, with the different foreign agency with this when we are doing nano applications. Then what is the government initiatives in nanotechnology in the agriculture? No doubt, government has to take a real nano mission, launch nano technology research centers like Center of Nanoscience and Engineering in the Indian Institute of Science, Bangalore, which was the nano fertilizer, nanotech food packaging. ICR initiatives is there on the both IRI, IVRI to develop nano biosensors are there, nano pesticides, nano capsules for nutrient delivery. This would be there. Then what is our way forward? So no doubt there is exciting result is coming with the potential is there as far the agriculture and animal husband is there. So future possibility. So multifunctional nano systems. The system can simultaneously improve crop yield, damage resistance, water efficiency, fertilizer places. So large scale network of nanosensors. This network will provide real-time monitoring of the soil, plant health, food quality through the supply chain. Smart nanopesticides, nutrients are responsive to the plant biochemistry for precise dosing and minimize toxicity. So these are some we will go for AI guided automation because now the age of AI, how the AI guided automation will enable data driven dynamics optimizations of the nanomaterial doses and applications. You can see how the nano means a dose or AI driven we should see enable nano coatings. So allow the self life and cleaning of this of the any of the particles. So finally, I can conclude that with the benefits like increased crop yield, animal performance, reduced wastage, and sustainability, nanotechnology can play a pivotal role in shaping future agriculture in India. So there is a lot of scope is there. We should know, we should do in a multidisciplinary approach how this nanoscience can be applied in both in agriculture, animal science, and in the food science system, so that our crop improvement will be there, animal will be there, environmental will be friendly, and so that it will be in and how the future of the nanoscience is really we can go with this. With this, I thank the organizer to give an opportunity to deliver with is there. Thank you very much for
Just thank you, lot, sir. So thank you a lot, sir. Am I audible? Participant, please.
Yes,
Yes.
sir, you are audible.
Yes. Audible.
Yeah. So right now it is open for question. Any participant wants to ask anything, any question? Please ask. Sir is here to reply. Please ask.
Good morning, sir.
Good morning. Yes, sir.
Sir, here it is Dr.
Doctor
Sir Aisha Mohanty.
Aisha. Okay. Yeah, from our university.
Sir.
Okay. Yeah.
Yes. Sir,
Yes, sir.
I have a question. Sir, as you said that, the long-term application of manure from this poultry diets may increase the soil toxicity. So sir, can we still use this, for long-term use, can we use this nanomineral supplemented poultry diets, and can it alter the diversity and function of the beneficial soil microorganisms? Like nitrogen fixers and VAM, mycorrhizal fungi or something like that, sir?
So you are telling Aisha poultry diet, you are telling, na? By giving in poultry diet, how it will apply to the manure and how it is coming to like this. Your
Yes.
question is like this?
Yes.
Very good. So one thing actually, you are very right. So perhaps in our experiment, we have not the availability we have seen actually in the serum it was very good. Huh? So your ask is it is coming through feces or like this you are telling, na? So that part we lack actually. We have not studied the feces excretion of the nanoselenium. So my idea is that when the bioavailability is more, just remember Aisha, when the bioavailability is more means it can absorb more, and we are giving a very low dose. So that's why the excretion pattern of the nano, whatever your nano is giving, is not that much. Like the other inorganic minerals, whatever giving it, phosphorus or calcium, the environmental pollution. Because if you see the observability and the availability of the most of the major minerals or the trace minerals is hardly 40 to 60%. So that's why rather that's rest 40 to 60%, or you are telling that is the how it is going to the environment through the feces and then polluting or the environment like this. But as far the nano is concerned, because you are giving a low dose and the excretion is somewhat not that much, not common. But your question is right. If this nano is penetrating, whatever is coming also, it's not 100% efficient. You see. So maybe that study is needed actually. Is there any toxic is coming to the soil or in the water level? That study should be done. That's why I'm telling now. It's not a very, by a simple experiment, we cannot give a very concrete conclusion. Still, study is required. So that how much, still some excretion is there. It's not 100% utilization is there. That has to be studied. It's a complete cycle. We should go for one more cycle actually. Then the environmentalist also involved, so soil scientists will involve, so that you can know what is its precipitation percentage in the soil and the water is there. Thank you.
Any other participant? Yes.
Sir, Pranam sir. Sir, myself Dr. Manas Ranjan Sanapati, Principal APC Supplement.
Okay, Manas. Yeah.
Sir,
Thank you.
sir, my question is
Yeah.
that you have prepared that green method which is in low cost. So,
Yeah.
that
Yes. Yeah.
preparation for pelleted feed in case of poultry.
So actually, what happens is we have prepared the nanoselenium, then it incorporated in the pelleted feed. Whatever nano is coming.
Sir, that is my question, that pelleted feed, you have used that
Yeah.
nanoselenium which is prepared by the
Yeah.
green method.
That actually, no, that we have not actually green method. That
Sir,
actually, because whatever green method is very economic.
Sir.
But the production means we cannot get whatever used by that time. We are using the inorganic method of nano. We are using
Sir.
that actually.
Sir,
And
if
the
you
recovery-
prepare through green method, then it will be more profitable, I think, sir.
Exactly. Actually, you are very right. We have just standardized actually. Green method is better one actually. So without even sodium selenate, we can use this actually. Right? Yeah.
Sir.
Yeah.
Sir,
Okay,
another
thank you.
question is that just like Madam Aisha has asked,
Yeah.
my
Yeah.
question just reverse, sir. As we know that in IFS system, all the components are interrelated to each other. If we
Yes.
are having that nano selenium or nano material, anything else, if it is present in byproduct of agricultural or crops residue, then we can use
Yeah.
it in case of livestock?
This thing can be used, you are telling, na? Same
Sir,
material you are telling. Na?
Sir,
That can be used, but that presence I am telling, na, whatever is nano, in that form, that may not be available, I am telling actually. When you are
Sir.
coming to excretion form, that form may not be suitable for what purpose you are using. Yeah.
Okay, sir. Thank you, sir. Thank you, sir.
Yeah.
Sir.
Okay, dear participants, and I must thank Sir for giving such a beautiful lecture. He said, as Sir has worked in this area, he shared a wealth of practical insight that added a great value to our discussion.
Thank you, dear participant, and thank you, organizer, giving me a chance for me. Thank you, Subhasri and Dr. Ediar also. Huh? You are present there. If anything
Yes, sir,
there,
yes,
you
sir.
can compliment us.
Thank you, sir.
Okay, thank you. I have another meeting at 12 o'clock. Yeah. Okay.
Okay.
Thank you.
Thank you,
Okay.
sir.
Yes. Thanks. Yeah. Full good. Yes. Yeah. Yes.
So, dear participants, welcome to this session, and the next session will start at 2:30 p.m. today. So, please join 10 minutes before. Link will be provided separately, sent separately. Thank you.
Thank you, sir.